Good morning, girls. Assalamu alaikum.
Good morning,
Miss.
How are you all?
Good morning, Miss.
Good morning.
Fine,
We're
Miss.
fine. How are you?
Alhamdulillah, I'm good. How are you all?
Alhamdulillah.
How was the exam today?
It was easy. Alhamdulillah.
Easy. Great. Which exam you had?
English.
English. Okay, great. Alhamdulillah. All right. We finished lipids yesterday and we are yet to do the lipid test, isn't it? Or we are yet to start the lipids. I finished with your class, right? Ladies?
We did the explanation for lipids, but we didn't do the tests yet.
Right, yes. Thank you so much. Yeah, so we'll we also finished from the textbook. So we'll do the test today for the lipids, and then we shall do the DNA, inshallah, and we'll be done with this topic. All right. So we have 10 attendees. So let's start. Do we have a request? Okay, Ari. Walaikum assalam. Okay, so let me quickly recap the contents we covered yesterday before we start the test. We shall also do about the test today, which in order to check the presence of lipids in the food. Give me a moment, ladies. Yeah. Okay. So the biological molecules. We'll start with Bismillah ar-Rahman ar-Rahim and Rabbi Zidni Ilma, because we did this verse many times. So objectives today are to conduct the or study about the tests which are done in order to check the presence of lipids or fats in the foods. So under the KWL, let me ask you questions about the topic we covered yesterday, which is lipids. Okay, let's start with the sources of lipids. From where do we get lipids? From the foods we eat? Ladies?
Dairy products, chocolates, ice cream, sweets, oils.
Okay, great. The various oils, the vegetable oils, also from the dairy products, and chocolates and sweets, of course, if we are using a lot of butter in them, yes. In chocolates, yes, because it contains nuts. If the nuts are present, then the chocolates, all of them, they contain fats. All right, very good. Fats, otherwise they are known as lipids. And what are the elements with which the fats are made up of? What are the elements, girls?
Carbon, hydrogen, and oxygen.
Thank you so much. Carbon, hydrogen, and oxygen are the elements with which fats are made up of. So when we break down one fat molecule, it breaks into how many parts, and what does it break into? What does one fat molecule consist of?
Glycerol.
Yeah, glycerol.
Miss.
And sweetie?
Fatty acids.
Fatty acids. Excellent. So each fat molecule, it is made up of 3 fatty acids and glycerol, as you can see in this picture over here. All right. So you can say if you're being asked about the composition of fat molecule, you will say it is made up of fatty acids and glycerol. All right, and glycerol, it is an alcohol with 3 hydroxyl groups. It is a thick, sweet tasting liquid which is dissolves easily in water. When it combines with 3 fatty acids, it becomes a fat molecule. And these fats, they are insoluble in water, of course, and fats which are liquid at room temperature are known as, what are they known as? fats which are liquid at room temperature are oils, isn't it? And the solids which are the fats which are solids at room temperature are known as fats itself, like the butter and the cheese and all that. The cream also contains a lot of fat. All right. So how does 1 gram of fat yield?
39 kilojoules.
of energy.
Very good. 39 kilojoules of energy, which is double the amount of energy which is released by carbohydrates, right? So, however, which is the first source of energy for the body? Is it carbohydrates or fats?
Carbohydrates.
Carbohydrates. And what is the use of fats in the body then?
They store energy
Heat.
and
Heat benefits.
Very good. The function of fats is insulation or providing heat to the body. Also, the amount of energy which is released by fats, it is stored as glycogen reserves so that our body uses it whenever we need excess or extra energy. All right. Of course, it provides the body with the energy, but it is not immediately used as carbohydrates are broken down for energy. But this energy from the fats, it is converted into the adipose tissue and stored under our skin. And also the fat reserves, they are stored as glycogen in our muscles and also in the liver so that whenever we need excess energy, we can use it. All right, so here in this table, you can see the comparison of carbohydrates, fats, and proteins. Do you want me to read out this table again, or would any one of you like to read it as a revision?
May I read it?
Go ahead, sweetie.
Elements. They contain carbon, hydrogen, and oxygen. Carbohydrates. Smaller molecules of which they are made are simple sugars and monosaccharides. Solubility in water. Sugars are soluble. Polysaccharides are insoluble. Why organisms need them? Easily available energy, 17 kilojoules. Fats. Elements they contain are carbon, hydrogen, and oxygen. Smaller molecules of which they are made are fatty acids and glycerol. Solubility in water, but they are insoluble. Why organisms need them? They are used to store energy, 39 kilojoules, insulation, making cell membranes. Proteins. Elements they contain are carbon, hydrogen, oxygen, and nitrogen. Amino acid. Smaller molecules of which they are made are amino acids. Solubility in water. Some are soluble and some are insoluble. Why organisms need them? Making cells, antibodies, enzymes, hemoglobin, also used for energy.
Excellent. Thank you so much, sweetie. So is it clear about the carbohydrates, fats, and proteins, ladies? Any questions? All
no,
clear?
thank you, Miss.
All right.
Miss?
Thank you so much. So let's talk about the food test now. The tests which are conducted in order to check the presence of the lipids in the food. Okay, so this is our presentation for food testing. Let me take you to the page wherein we are doing the tests for the lipids. So this is a Biuret test which we spoke about the last time, which is conducted for the presence of proteins in the food, and this is the test for the fats. Okay, so the name of the test, it is known as the emulsion test. So the emulsion test, it is a method to determine the presence of lipids using wet chemistry. Okay, so what do you understand by wet chemistry? Give me a moment, sweeties. Alright, so what is the test known as? It is the emulsion test. It is a method to determine the presence of lipids. The procedure is for the sample to be suspended in ethanol, allowing lipids present to dissolve. And the lipids are soluble in alcohols, and the liquid alcohol with dissolved fat is then decanted into water. You may also check in your textbook Do we have it? Yeah. So on page number 45, you have the testing food for fats. Can you see it, ladies? So you can see this picture of a test tube which is shown a positive result for the emulsion test. Emulsion is basically submersion into water. Okay, meaning emulsion of the fat droplets in the ethanol or water mixture is clearly shown. Okay, for example, we are taking an oil and testing whether it contains fat or no, because we are taking any food which contains fat. So that if you are testing along with this ethanol, then if you see the emulsion in it, meaning if you see the presence of fat in it, it will turn into a milky white color. Then you will see the presence of fats in it. Okay, so like this, as you can see. So emulsion test, it is a method to determine the presence of lipids using wet chemistry. Why are we saying wet chemistry over here? Is because we are using ethanol. Okay, so have you heard of the term ethanol, ladies, earlier?
Yes.
Yeah, what is it, ethanol? Can we say it is a kind of an alcohol?
Yes.
Yes, it is a kind of an alcohol. Ethanol is used in order to test for the fats. That is why we say it is using wet chemistry. Okay, so the procedure is for the sample to be suspended, meaning the oil to be suspended in ethanol, allowing lipids present to dissolve. The lipids are soluble in alcohols. Okay, so if we are using water instead, the fats, they are not soluble in water, isn't it? So clearly you can see the oil floating on top of the water. That is why we need to use the ethanol over here. Why? Because the lipids, they are soluble in alcohols like ethanol. So the liquid, which is alcohol with dissolved fat, is then decanted into the water. Decanted meaning whenever this fat, it dissolves into the ethanol, it shows like this, like an emulsion clearly separating from the water, okay, showing the presence of the fat in that particular food. Okay, so the procedure is for the sample to be suspended in ethanol, allowing the lipids present to dissolve in it, as the lipids are soluble in alcohol. Then the liquid alcohol with dissolved fat is then decanted into the water, meaning it is suspended like this, okay, without getting dissolved. Excuse me. So this is how you test for the presence of lipids. Okay, so for this test you can use the vegetable oils, any vegetable oil which is available, like the corn, groundnut, or even olive oil, or you can also use the fats like the butter, or also the cream, not the cream actually, butter only. Okay, so these you can use in order to check for the presence of lipids in the food. All right, ladies, is it clear about the test for the lipids? Any questions? What is the test known as?
Emulsion test.
Yeah, it is known as an emulsion test. So what is used in this emulsion test in order to test for the presence of fats?
Ethanol.
Ethanol is used, which is a kind of an alcohol. So as ethanol is used, we are saying this test, it is using wet chemistry, because ethanol, it is a kind of a chemical which is alcohol. All right, so let's move back to our presentation now. All right, even in this presentation, the various chemical tests for biological molecules are again repeated as a kind of a revision for us. So all living things are essentially composed of the same basic group of molecules. So the four types of carbon-based molecules that are carbohydrates or starch, proteins, fats or lipids, and also vitamins. Okay, so the scientists often need to know whether or not a particular type of molecule is present in a solution, for which we need to do the various food tests which we spoke about already. For example, a doctor might try to detect glucose in a urine sample. Why does the doctor have to detect the presence of urine, ladies? As I told you earlier.
For diabetes.
Very good. For diabetes, because mostly the patients, they lose the glucose in their urine, the diabetic patients. So in such a case, the doctors, they have to check the sample of the urine to detect the glucose, the presence of glucose. So glucose in the urine suggests the patient has diabetes, or an environmental scientist might test for the starch in the outflow from a food factory. Okay, so there are a number of simple chemical tests that can be carried out on biological solutions. The testing for fats first, so the testing for the lipids. An important feature of fats and oils is that they are insoluble in water. This means that you cannot make an aqueous solution of a fat or oil on which to carry out a biochemical test. So what do we do now? However, the fact that lipids are insoluble forms the basis of a physical test. So as the fats, they are insoluble in water, we cannot mix them in water or conduct any test like we did for the proteins and for carbohydrates. So we need to use some other thing in order to check the presence of fats. So such a test is known as the emulsion test. This is known as the emulsion test. 2 centimeters of ethanol are added to the unknown solution and the mixture is gently shaken. Okay, the mixture is poured into a test tube containing an equal volume of distilled water. Okay, so this is the test tube which he is pouring into. This contains what? It contains ethanol, and along with that you can use anything which you're testing for over here, maybe oil or butter or anything. You shake it well, and this mixture you pour into another test tube which contains an equal volume, as much as volume you're taking of the ethanol over here, of the distilled water. So if you're adding this, if the solution or the lipid which you're using, in that solution, if the fat or the lipid is present, it turns into a milky white emulsion, as you can see. So meaning, along with the ethanol over here, any vegetable oil has been mixed, shaken, and added to this distilled water. So when you see this milky white emulsion being formed, it proves that this particular oil or anything which you're using over here, it contains the lipid. Okay, so this is how you test. The test is known as an emulsion test, which is conducted in order to check the presence of lipids or fats in the food. Okay, and this test, it is known as the test which we do for proteins. What is the name of the test, ladies?
Biuret Test.
Biuret Test. Excellent. So what do we do in this test? Can someone please read out? Of course, we did already.
Half fill a test tube with the extract you wish to test for protein. Add 5 to 6 drops of Biuret solution. This solution contains copper sulfate solution and sodium hydroxide solution. Safety. Take care as sodium hydroxide solution is corrosive. Biuret solution usually looks blue in color. A positive test for protein is if the Biuret solution turns purple, violet, or lilac. If the color remains blue, then the extract does not contain protein.
Excellent. Thank you so much, sweetie. So the test conducted for the presence of proteins, it is known as a Biuret test, wherein you use a Biuret solution, which is of color blue. Biuret, it is actually copper sulfate solution and sodium hydroxide mixed together. So whenever you're adding the food containing protein to this Biuret solution, if it remains blue, meaning the food, it does not contain protein. But if it turns purple or violet or lilac, meaning the food contains protein. The deeper the color of the solution, the more the peptide bonds which are present in the protein. So that was about the protein test. Now who is going to talk about the iodine test for starch? Read out from the slide here.
When iodine solution is mixed with starch, it turns from brown to dark blue. As this happens with very small amount of starch, iodine test is a sensitive.
Very good. So you can see the test over here clearly how they are doing this test, ladies. So here you can see the various foods containing starch, okay, or carbohydrates like the cookies, French fries, potatoes, oats, rice, wheat flour, etc. Okay, so you can see what happens next. This iodine solution, it is dropped into a potato slice. Then you can observe what happens next. So what is it changing to, the color?
Brown.
brown. So you can see it is being added to various carbohydrate sources over here, to a slice of bread. And what was the second one he added that to? C. The change of color, meaning the carbohydrate is present. Similarly, you can see the nuts over here. The Biuret solution is being added. And okay, what is in the, this is nuts. What was the first one? What is this, ladies?
Is it oil?
It is an oil. Yeah, so does it contain any carbohydrate or starch in it? No, of course. That is why it will not change color. Whereas the bread has changed color, you may see. And also the nuts over here, what do they contain? Do they contain carbohydrate or protein? The nuts, they contain protein. That is why you can see no color change. Similarly, the oil, it has also not changed the color, the iron, because it does not contain starch in it. So whichever foods contain carbohydrate, in that if you're adding iron, it will change to brown color. Okay, brown to dark blue. All right, so that is about the test. This is the glucose test. Who's going to talk about it?
To test for glucose, a reducing sugar 2 centimeter cube of
Benedict's reagent.
Benedict's reagent are added to 2 centimeters cube of the unknown solution, and the mixture is heated in a boiling water bath for 2 to 3 minutes. An orange brick red color is a positive result. Glucose is present.
Very good. Okay, so this test is otherwise known as Benedict's test, in which you can see Benedict's reagent is used. Okay, so if the glucose is present, it starts changing color from green, yellow to orange, and finally to brick red, proving the presence of glucose in the test. So you can conduct 2 tests for the presence of carbohydrates. One is known as the starch test, which is the iodine test, to test for the presence of starch, whereas test the presence of glucose, which is known as a Benedict's test, in which a Benedict's reagent is used. Clear about all the tests, ladies? Any questions?
No, miss, thank you.
You're most welcome. So let's talk about water now. So we already studied that water is also an important component, not only in the human body, but we need also to drink water. Water has various uses, isn't it? So most cells, they contain about 75% water and will die if their water content falls much below this. So water, it is a good solvent, and many substances move about the cells in a watery solution. Many important metabolic reactions also take place in aqueous solution, and the 2/3 of this water is in the cytoplasm of the cells, and the 1/3 it is in the tissue fluid in the blood plasma. So we already studied that most of our body is made up of water. 2/3 of the cytoplasm of the cells contains water, and even our blood plasma, it is entirely made up of water. So a loss of only 5% of the body's water can lead to unconsciousness, and loss of 10% would be fatal. That is why dehydration, it can be very, very dangerous, ladies. So whenever the young babies or children, they have dehydration, even the adults, if you have continuous watery diarrhea and also the vomitings persisting for more than 3 days, you are immediately admitted and you are given saline, which is glucose. You are given this, what do you say, intravenous glucose is given to you. Why? Because you lose water due to dehydration. Okay, so it can cause a lot of effect. If you're losing 5% of the body's water, you can get unconscious, and 10% could be fatal as well. All right, so that important the water is. You cannot survive, you can survive without food for about till 3 days, but you cannot survive without water for more than a day. You must have very well experienced it when you observe fast during Ramadan, that suhoor till iftar, you don't want to eat, but you first want to drink, isn't it? You drink a lot of water and juices in order to quench your thirst first, and then you go towards the food, right, ladies? So water is very, very important. Also, for the transport of nutrients, example glucose and amino acids in blood and sucrose in the phloem. So removal of excretory products like ammonia, urea, secretion of substances like hormones and digestive juices, all this happens because of the water. So water plays a major role in various metabolic reactions which take place in the body. All right. So that is about it, ladies. So we don't have the information about the nucleic acid for deoxyribonucleic acid, which is DNA. Secretion of substances like hormones and digestive juices, all this happens because of the water. So water plays a major role in various metabolic reactions which take place in the body. All right. So that is about it, ladies. So we don't have the information about the nucleic acids over here, the DNA. So let me also complete about the DNA from your textbook so that we are done with this topic. How much time is left, ladies, according to the new schedule? Do you have it with you?
9 minutes.
9 minutes. It's still 11 o'clock, right? Okay.
Yes.
Okay. So I want you all to open page number 47 from your textbook about the DNA. Okay, so we shall talk about DNA now, which is the last page of this topic, and then we are done with it. So DNA, we already know it is the genetic material present in the nucleus of the cell. So what else do we have to learn about DNA? Why is it as important, or why is it known as a biological molecule? Stands for deoxyribonucleic acid, which is DNA. And DNA is the chemical that makes up our genes and chromosomes. Right? So that is the genetic material in our body. It is the material that we inherit from our parents, which gives us many of our characteristics. Okay, so that is what as genes is known as. So we inherit our genes from our parents, isn't it? So all the physical characteristics chromosomes from our parents. You either resemble your uncles or your paternal aunts or uncles, so which other compared to your mother, and sometimes you are a mixture of inherit this. You pass on this genetic information or the DNA to your offsprings for the future generations. So you can see in the figure on your page number 47, it is the figure of a DNA as to how it is a twisted ladder structure. That is how it is the double helix. That is how you can explain a DNA molecule. So it shows a structure of a very small part of a DNA molecule. It is made up of two long strands, each with a series of bases arranged along with it. The bases on the two strands are held together by bonds, forming cross links. And the two strands then twist together into a kind of a spiral called a helix. Okay, so this helix, it is a twisted ladder kind of a thing. And what do you see in these? You can see the arrangement of these bases. What are the four bases which are present in the DNA molecule, ladies? They are the adenine, guanine, cytosine, and thymine, right? So these are the four bases which you can see. Sorry. So this is how the structure of a DNA molecule looks like, isn't it? So this is the double helix structure of a DNA molecule, and this is what you have in your textbook as well. And here you can see the bases which are arranged like this. The adenine, the guanine, the cytosine, and thymine. Because of the different arrangement of these bases, each and every DNA has a different structure, and each and every protein molecule has a different arrangement of these bases. All right. There are four kinds of bases known by the letters A, C, G, and T. As I told you, these letters which you can see, the A, G, C, and T, what are they? They are, if you look carefully, you will see that T and A always link up with each other, and also C and G. The bases, they always pair up this way. So C, cytosine, is always linked with G, guanine, in this arrangement, and A you can always see linked with T, which is adenine and thymine. Okay, so this is how you can see the bases are linked. Can you see it clearly in this picture, ladies? Give me a moment. Okay, here, can you see it? So you can see the A always linked with the T over here, and C with the G. So this is the basic pairing, though sometimes it can be a different pairing depending upon the protein molecule. But basically the basic arrangement is always you can see the combination of A and T like this, and the combination of C and G like this. Can you see A and T base pairs and C and G base pairs linked together like this in the structure of a DNA? Alright, so the sequence of the bases in our DNA, it provides a code that is used to determine the links of the proteins that are made in our cells. Okay, so this in turn determines how our cells, tissues and organs develop. So each and every protein molecule, it has a different combination of these base pairs, also a different combination of the way these base pairs are arranged in the structure of a DNA, because this makes an amino acid, and you can see the various combination of amino acids in a protein molecule. And of course all your tissues and organs and cells that are made up of these proteins. So this is how the structure of each and every organ in your body is made, depending upon the sequence of these bases. The sequence of the bases in our DNA, it provides a code that is used to determine the kind of proteins that are made in our cells. This in turn determines how our cells, tissues and organs, they develop. So the sequence, it determines that you are a human and not a tree, because it is a different sequence for a human, for various animals, birds, fishes, and also for the trees, the living things. Okay. As well as many of your personal characteristics such as your hair color and your blood group, all this is decided by this arrangement of the bases. Okay, so you will find out more about DNA as to how this DNA molecule functions and how are these bases arranged. You will study further in your following topics. For now, this is the information you have about the DNA. Clear, ladies? Any questions so far? Any questions regarding the DNA molecule and the arrangement of the base pairs? Yeah.
No, thank you. All clear.
You have a question?
No.
What are you saying? What were you saying, sweetie? You're saying 2 minutes are left? I don't know. Okay, I'm downloading the attendance. 23 girls are present. Can you please repeat, because I didn't get what you said. Okay, she says no thank you, all clear, Ravasan and Arij. Okay, so can you please tell me what are the 4 bases which are present in the DNA molecule structure?
Adenine, guanine, cytosine, and thymine.
And thymine. Excellent. So which two always pair up together? Which 2 bases?
Adenine and thymine and guanine and cytosine.
Very good. Adenine and guanine, they pair up together, and cytosine, adenine and thymine, they pair up together, and cytosine and guanine, they pair up together. So what is the full form of DNA?
Deoxyribonucleic acid.
Deoxyribonucleic acid. Excellent. And how do you describe the structure of a DNA molecule?
Like a ladder.
Double helix. Yes, you can say double helix or a twisted ladder. It is a structure of a DNA. Okay, great, ladies. That is about this topic. I want you all to complete the homework. I was usually not assigning because you have your midterm exams. So whenever you're done with the exams and you're free, I want you all to complete the questions for 4.5, which is DNA. The end of the chapter questions, of course. And did you complete the questions for 4.4 proteins, ladies?
Yes.
Yes, you did till proteins, right? So you are yet to complete the questions for fats, which we did today, and also for the DNA and the end of the chapter questions. Okay, so no hurry at all. So whenever you're free, when you are done with the exams, I want you all to complete all these questions and submit. With this, we are done with this topic, ladies. Any questions so far? Anything, any more information you want? Okay, I'll send you the notes for this topic. I'll upload it on Teams. Okay, thank you. Then you have a great day.